Thank you. I hope you can see me behind the podium. I'm not very tall. I am Lida. I'm a PhD student under Eva Maria Novoa at the Center for Genomic Regulation, and today I will be presenting my work on how we can chart the tRNAome in health and disease with AMaNITA. So in high school biology, I think we were all taught that tRNAs are those little molecules. They grab an amino acid from somewhere in the cell, they bring it to the ribosome, and they help with the synthesis of the polypeptide chain, and that's it. Thank you, that's my talk. No, actually, even though we have been regarding them as this very stable entity that is very central to translation, they are actually way more interesting, and this is why I have a PhD. Now, so tRNAs are the most abundant RNAs in the cell in terms of expression. They are very short, typically 70 to 90 nucleotides long, and they are very heavily post-transcriptionally modified. Those RNA modifications affect translational dynamics directly by either affecting the tRNA structure and therefore the function of the tRNA, or by affecting the tRNA-RNA interactions. And the dysregulation of the tRNA abundance, so the expression, or the dysregulation of the tRNA modifications have both been linked with involvement in mitochondrial diseases, neurological disorders, or even cancer. Now, as you can see in the little image on the right, the tRNAs, as I mentioned, are very heavily post-transcriptionally modified, but they also carry a ton of different RNA modifications within the same molecule. So the question then becomes, how can we accurately detect RNA modifications, especially if it carries so many same, or different RNA modifications in the same molecule? The answer so far has been short-read RNA sequencing with two different types of protocols, either antibody-based or chemical treatment-based. One of the most popular chemical treatment-based is bisulfite sequencing. I'm pretty sure you all know this. This is how we study DNA methylation. However, both of these protocols are very limited to be modification specific. At best, they can capture a couple of modifications in the same molecule at the same time. So we are missing the layer of capturing the diverse modifications found in a single molecule. And here comes Oxford Nanopore direct RNA sequencing with the promise that when you sequence an RNA molecule, you don't get any bias, you are just sequencing the native sequence, and you're also sequencing the native modifications. In this frame, we have single cell molecule resolution, we have isoform-specific RNA modifications, and we can also capture the stoichiometry of the modifications, the percentage of reads that are actually modified. In principle, we can also capture all RNA modifications, which is particularly useful in studying tRNAs when we have so many diverse modifications. And again, in principle, it requires no custom protocols. The reality is a bit different. So imagine this is an RNA molecule passing through a pore. It generates a little intensity squiggle, which the base caller was trained to base call into the 4 canonical bases. Now, ideally, what would happen is that when a modified molecule passes through the pore, here denoted in orange, and the base caller would identify the modified residue, the modified base itself. This is the idealized case. This is the promise of direct RNA sequencing. We are sort of here. So the base caller is expecting one of the 4 canonical bases, especially if it's not modification aware. So it makes mistakes. So it has to make a choice between 4 cases. You're not giving it a 5th. So it makes errors. We call those base calling errors, and we use them as a proxy for modification information in direct RNA sequencing. And this is essentially what I will be working with in this presentation. So moving on to how we can accurately capture tRNAs. As I mentioned, direct RNA sequencing is very promising because we can capture both the sequence information and the modification information. In this case, Nanopore, the technology itself was very biased towards longer reads. So all of the short reads, and tRNAs are particularly short, were discarded. So there was a need to create a custom protocol in the end that can accurately enrich for the tRNAs, and we can sequence them. Some of the improvements that were known, that were introduced, they were built upon earlier protocols by Thomas et al., which was published in 2021. And they optimized the library protocol to increase the yield, and it also provided quantitative RNA modification information. Until that point, it was mostly qualitative. This was a protocol developed in our lab by Morgan and Leszek, and all of the data that I will be presenting today is using this protocol to generate tRNA libraries. So nano-tRNA-seq provides the basis for differential tRNA expression and differential modification analysis. On the left, you can see that we can use it to identify differentially expressed tRNAs in yeast. This was an experiment from the protocol paper. We were seeing differences under different stress conditions. And on the right, you can see an IGV plot, and we see how these base calling errors that I was mentioning before show up as a signal there at position 55 for pseudouridine. However, we were missing a statistical framework that could tie all of this together, give us some p-values, give us some clarity and some security that we're actually capturing some real biology. And this is my project. It's called AMaNITA. It's a good name. But it also stands for Abundance Modification and Nanopore Intensity Toolbox/application. Where does it come in? It comes at the very, very end of your analysis. So if you've done your library preparation, you have isolated tRNAs with the nano-tRNA-seq protocol, you sequence it, you base call it, and then you want to interpret your data. AMaNITA comes in, and you can perform quality control, batch effect correction, estimation and correction, and then perform differential abundances and modifications analysis. Briefly, we acquire the library from tRNA-seq protocol, and then we base call it and align it. And then AMaNITA works with very simple input. It works with the aligned BAM files and the metadata, and also it requires the information about the organism you're working with and also the tRNA group, whether you're working with cytoplasmic or mitochondrial tRNA. In general, we are suggesting those two populations are analyzed independently because they behave very differently in the cell. And we have introduced several modules that perform different tasks. One is filtering, that performs filtering. Technical, which performs quality control. The batch module, which performs batch effect investigation and correction. And two pairwise modules, the simple one and the more custom one, which do pairwise differential analysis, both for abundances and the modifications. And all of this, depending on what the user chooses to run from those modules, are concatenated into a single AMaNITA report. Now, we had to benchmark it, and especially we had to benchmark it to make sure that we are accurately capturing RNA modifications. For this, we had 7, I will be showing 6 of them, knockout strains in yeast. We had 7 different knockout enzymes. Those enzymes are responsible for the deposition of a specific modification in the tRNA molecules. I will be showing a specific type of heat map from now on, so I will take the DUS1 knockout as an example to explain it. This is a structurally aligned heat map. The x-axis is the position, the y-axis is, so essentially each line is a tRNA, and those tRNAs are aligned so that we are retaining the structure of the tRNA. The gray boxes are positions that are not present in specific tRNAs. So for example, for this DUS1, we were expecting, DUS1 is responsible for the dihydrouridine modification at position 16 and 17, and we are seeing this. DUS2 is responsible for, again, dihydrouridine at position 20. We are seeing signal in this position. DUS3 at position 47, again, for dihydrouridine, we're seeing some differentially downregulated modifications at this position. TRM1 is responsible for M22G at position 26. We are seeing signal at this position. TRM8, it's an M7G at position 46, and TRM10 is an M1G at position 9. And we see that for all knockouts, we are getting a signal at this position. But you might also be noticing that we are getting signal a bit in the flanking regions, in the neighboring positions. This is a current limitation in the field in general. So as the nanopore as the RNA passes through the pore, the current it creates, especially if it's a modified base, might affect downstream positions. And I'm saying downstream because direct RNA sequencing happens 3' to 5', so in this direction. So essentially, as you can see, all of the affected neighboring positions are 5'. This is a limitation that we have to have in mind when we are analyzing this type of data. However, this is the best that we've currently got in the field. But it's not just yeast. We also benchmarked it against other species. This is a wild type experiment in mice. Here we knocked out QTRT1, which is responsible for the cues in modification at position 34, and DNMT2, which is responsible for the M5C at position 38, but at specific tRNAs. Now, I should note at this point that about a year ago, a new nanopore chemistry was introduced. It's RNA 004. However, many people have been working with the previous chemistry, which is RNA 002. We wanted to make sure that if you have data from the previous chemistry, it will not go to waste, and that this tool is robust across chemistries. It is. On the left, we see the QTRT modification. We were expecting essentially 4 positions to be modified. You have to believe me that those are the actual 4 tRNAs that are modified. And again, we are seeing that the signal is spilling a bit on neighboring positions. And then in the case of DNMT2, we are seeing the signal at the expected tRNAs, but we're seeing them in the neighboring positions. And again, it goes to show how it can be a bit spilling over and causing some differential events in nearby positions. And our next step was to apply AMaNITA on a non-benchmarking data set. For this, we had mice tissues from 8 different tissues and across 2 diets. In this experiment, we wanted to check whether the tRNAome, the tRNA population changes between tissues, but also between diets. We had approximately 4 replicas per tissue and diet, so statisticians should be happy about that. Now, So in our first look of the dataset, it looks like our data mostly segregates based on tissue. However, upon further inspection, we noticed that sequencing batches and library preparation steps introduce very heavy batch effects in the dataset. Here I am showing the PCA plots on tRNA abundances only. On the left is based on the RNA extraction date, and the second is the run ID, or the sequencing date, essentially. And we see that there is very heavy effect of both of those batch effects that are explaining the variance in the data, especially in the PC1. So this is something that it's better to have in mind and take into account when a data set is analyzed, and this is why we wanted to introduce the batch module into AMaNITA, so that this is automatically investigated and also automatically adjusted for, or if the user can know that you have this batch effect, maybe next time take it into account. And this is our data after batch effect correction. So adjusting the batch effect correction leads to cleaner segregation, of course, and it also shows us exactly how the tissues are clustering better. Interestingly, in both abundances and modifications, brain cluster with testis, and then the rest of the tissues are a bit on their own. And it's quite interesting to us because the modification signal is quite prominent. I will not go into more details about the results of this data set. I just wanted to use it as an example of why we need batch effect correction. But we are seeing actually that what we're seeing here in the segregation based on modifications, we are actually capturing differential positions between tissues and between diets. So I'm skipping that because I want to go to something I think a bit more interesting. We wanted to apply AMaNITA to a clinical data set, and we were lucky to collaborate with people from the Heidelberg University Hospital. We had a lung cancer cohort of 69 matched tumor and normal samples, and the initial segregation seemed good. However, again, we were seeing a lot of batch effect. So again, our batch model seems to be useful here. So adjusting for the batch effect, of course, leads to a better segregation of the groups, and it shows us that the tRNA for selenocysteine is the one driving the segregation, and it's more expressed in normal tissue. And when we did differential expression analysis, where we're actually catching this tRNA as differentially upregulated in the normal tissue. So to put it in a different way, the tRNA for selenocysteine seems to be downregulated in lung cancer, which is interestingly consistent with some clinical studies from the 90s, which were saying that selenium selenium-rich nutrition could help cancer patients with their chemotherapy. Now, to conclude, we have built a tool, AMaNITA, which captures differential modifications when benchmarked against graph truth datasets. And it can also inform users about batch effects and automatically handle them. When applied to non-benchmarking dataset, we can get insightful biological information, for example, about tissues or diets. And when applied also to clinical cohorts, we can identify tRNAs as potential diagnostic or therapeutic targets. This is the end of my presentation. I would like to thank my lab, Laia and Alexandra, who designed the experimental procedures, and all of you for the attention. I'll take any questions. Thank you.
Questions?
Are some of the sites fractional, so they're not present in all tRNAs?
I'm sorry?
Are some of the sites fractional?
So tRNA fragments, you mean?
No, I mean like one site for one specific tRNA. It's not modified in all of that.
So far we don't have very clean stoichiometry of the modifications, if I understand this is what you're asking, but we are getting them, and we think that with this approach we can actually find the percentage of reads that are modified or not.
And then
this
you
is
can
currently
check if it switches between tissues or things like that.
Yes, this is something that we really want to see, but we're not exactly there yet, neither in the technology of direct RNA sequencing, and also this is something that I'm currently exploring in terms of benchmarking the tool.
And then do some of the sites depend on each other?
Oh, yes, totally. Yes, this is true.
Hi, thank you. There have been some methods proposed that detect RNA modifications directly from the squiggle data rather than from the base calling errors of nanopore data. Have you tried those or do you think it would make sense to use them for tRNA? Would that be applicable? Have you tried it? Any opinions?
Thank you. This is a fantastic question. This is something that we've mostly applied in the lab with mRNAs and rRNAs. tRNAs are very noisy, so it's not very easy to distinguish the actual intensity and the differences in intensity. However, this is something, and this is why this is in the name of my tool. This is something that I actually want to incorporate at some point. But we have been seeing that it's a very noisy signal to try to deconvolve. So for now, base calling errors are our best bet. Thank you.
I'm gonna win.
Is it available?
It's not available yet. We're currently preparing the manuscript, so it will be available as soon as the manuscript is out. Remember to start it. Thank you.
Okay, thank you very much.
Thank you.